Good afternoon, everyone. Welcome to the GLAZE webinar series. My name is Neil Matson. I'm a faculty member at Cornell University and one of the co-PIs with the Greenhouse Lighting and Systems Engineering Consortium. We welcome you today to our webinar series, and it gives me great pleasure to welcome a good friend who's Ricardo Hernandez. Ricardo is an assistant professor in horticultural science at North Carolina State University, where he leads the Controlled Environment Horticulture Lab. Previously, he received his PhD and worked as a postdoc as well at the University of Arizona. Today, he'll be speaking to us about the importance of light quality in CEA systems. And if you have any questions during his talk, please use the question and answer box that comes up. We'll save most of the questions to the end, and I can respond to any like logistical or AV questions as they come up in the Q&A as well. With that, let me invite Ricardo to take the floor. Hi, Ricardo.
Hi, Neil. Thank you very much for the introduction. Please let me know if for some reason I lose audio or if I'm not being explaining myself correctly, okay? So thank you very much, Neil. A little bit about myself. As Neil mentioned, I lead a group of research at NC State University. We have several funded projects, a lot of them focus on light, intensity, quality in the interaction of light with other environmental conditions. And today I'm going to talk to you about the importance of light quality for controlled environment systems. And I decided to make this presentation two parts. The first part is just a very general introduction about the general impact of light on plant growth. And then after that, I'm going to move into some examples on how we have used that general knowledge to try to create some impact on the production of plants in controlled environment agriculture. So before we talked about light quality, I just want to reiterate that light intensity is probably one of the most important components for plant growth. And as most of you know, daily light integral, which is the accumulation of photons in a given area for a 24-hour period, is directly correlated to light. For most crops, especially for those that are horticultural crops, 1% increase in light typically equals 1% increase in yield, right? Now the slope of increase may be different, but typically there is a linear response to light, especially in the natural environments. But now let's move to light quality. Here you have a scan. This is a spectral scan of the sun radiation. And the units here is micromoles per meter square per second per nanometer. So basically, when you have the impact of the sun in terms of photons, we have a very, very balanced spectrum. That's why we call it white light. The percentage of blue, green, red, and far red is a little bit different, but overall pretty balanced. Here on your x-axis, you have the wavelengths. So basically, from 400 to 500, you have the blue light. From 500 to 600, we call it green light. It can also be considered as a yellow light closer to 600. And 600 to 700 is the red light. And 700 to 800, we have the far red light, which is a unique term for plant biologists. If you ask me to summarize the responses to light quality on one slide, I'll probably try to show you this slide. This is an oversimplification of the impact of light quality and colors to plant growth. Here in the wavelengths of UVB and blue typically create lower leaf area. Also anthocyanin concentration is promoted. Also a more compact plant overall can be produced with higher levels of low light. Then you have between the 400 and 700 nanometers, which is typically considered as the light used for photosynthesis. Now with more recent research in the past 2 years or so, we know also that far red light has an important contributor towards photosynthesis. But for general explanation here, we can consider 400 to 700 has the main wavelengths to increase plant growth through photosynthesis. Then you have far red light that by itself doesn't mean a lot, but together with red light, or the ratios between red and far red light can elucidate other plant responses, such as the increase in leaf area, the increase of plant height. Is also very useful for the perception of day length. Now there is, if we look at the sun, the impact of different radiation spectrum on plant is quite diverse. Again, you have 400 and 700 nanometers for being the photosynthetic active radiation for photosynthesis. Then you have the ultraviolet radiation, typically divided into UVA, UVB. You know, UVA radiation is typically absorbed by the same photoreceptors and your blue radiation. UVB can be also being absorbed by your cryptochrome photoreceptor in the blue, but also it has its own unique photoreceptor. Then you have the infrared radiation, which is greater than 800 nanometers. That, generally speaking, is nothing but heat, you know, radiative heat. Then you have your red and far red radiation, which is the red between 600 and 700 nanometers, and the far red between 700 and 800 nanometers. And the ratio between those two elucidate different plant responses. I took this scan from Tace and Seeger, a plant physiology book, the latest edition. And you can see how there is different pigments and photoreceptors that have a specific absorption peaks and a good spread of absorption of the wavelengths. So all of these photoreceptors, or a version of the photoreceptors, are present on plants. Therefore, the plants have developed a unique and very advanced a photoreceptor system to change their growth, architecture, and development based on light quality and light colors. Here, you know, is the same figure with some divisions of the photoreceptors. You have the UBR8, which is a photoreceptor that has been discovered to exclusively absorb UVB light. Then you have photoreceptors that absorb light between UVA and the blue, which are phototropins and cryptochromes, the most studied. And then you have another photoreceptor, ZTL. And then you have the phytochrome, which basically absorbs in the light in the red and far red portion of the spectrum. These are just general responses to blue light. You know, one of those is phototropism, right, which is when the plants are following the direction of the light. Here you have an example of a plant, a wild type plant with a source of blue light to the right, how the plants are moving towards the light. And then you have that same plant, but with a mutation on the gene phototropism that controls the phototropin, and the plants are not responding to the blue light because that gene is no longer present. You have leaf positioning, chloroplast accumulation, and overall stomata opening. And again, these are general examples on plant responses to blue light. Here, this is an old study now that we performed a few years ago, and you can see how the impact of blue light is decreasing the plant height of this young cucumber seedling. Is 10% blue, cool white fluorescent, which lands around 20% blue, and you increase in the blue light, you make it the plant more compact. But also, when you remove red light completely from the equation and you give 100% blue light, the plant response is different. So what I'm trying to get with this slide is that it all depends, and the ratios are very important. Now, general plant responses to UV light, you know, and UV light can be divided between UVA and UVB, 320 nanometers to 380 nanometers UVA. And UVB, which is actually what gives you a tan, is between 280 and 320 nanometers. You have other UV like UVC, but typically that's for germicidal uses and is not part of the sun spectrum. Now, the UV in general has similar responses to UVA, to blue, which is decreases the shoot height, decreases leaf elongation, decreases leaf area. It can also increase the thickness of the leaf. You know, it can create more branching. And sometimes, if the levels are very high, it can also lead to a reduction in growth. And actually, if you look through the literature and try to compile the majority of the studies under UV light, the amount or the intensity provided in those studies typically is several times higher than the one present in the environment. So a lot of them agree that high levels of UV actually reduce plant growth. But when you use intensities more closer to the outside environment, there is not a quantified reduction in growth, right? But instead, a good change in morphology, such as, a more compact plant as well has more, for example, phytochemical production. Green light in general, most green light responses, um, uh, uh, especially to the ratio, are more noticeable on the lower light intensities. And also higher green light triggers shade avoidance responses. And for those of you that are not familiar with shade avoidance responses, I think this acronym is perfect. Shade avoidance, right? So the plants are trying to avoid the shade, typically leads to plant stretching and also leaf area stretching and to try to reach the light, similar than those that are um, elucidated by far red light. Another common knowledge about the responses to green light, typically the green light serves as a slow down or stop for blue light responses. If you have a spectrum with high amounts of blue light and you, generally speaking, introduce green light to that spectrum, then the original response to blue light will be less. As an example, if you have a very compact plant growing in a very high blue, if you introduce blue, the plant will be less compact. So it serves as a slow down or stop to blue light. This is green light. Another one, which is the most studied photoreceptor here, is the phytochrome response. So basically, we have a protein in the plant that changes its isomeric state when it receives the red or far red light. Typically, PR is the phytochrome inactive form. And if you put red light into the PR, then a portion of those proteins actually become PFR, and that leads to cellular response. One example of that cellular response may be a stop stretching. And then during the darkness, then it returns back to the inactive form. So you can use red and far red light ratios to change the pool of these proteins within the plant. And they absorb in the PR absorbs mainly in the red, and the PFR form, once is the isomeric form, absorbs mainly in the far red. But they also have absorption peaks in the blue. And one of the most known responses to the ratios of red and far red is the shade avoidance response. So basically in this graph here, you have again that same spectrum that I showed you initially of the sun, which is very balanced in all the colors. And then we took the same spectrum, but inside the canopy, right? So in the middle of the canopy, you can see that most of the light in the blue, most of the light in the green, and a lot of the light of the red is absorbed by the plant. And what's left in the canopy, you have green light and you have far red light. So the proportion of green light and far red light inside the canopy is greater than other colors, right? So that's why a young plant in the middle of the woods, shaded by taller trees, will have the main spectrum that plant will be exposed is a green and far red rich spectrum, which will create a shade avoidance response and consequently stretch. So those very general responses help us plant scientists trying to understand that knowledge and create some value for production. And I'm going to walk you through a few examples of projects that we have worked on that present this value. The first one is using the dynamic control of light to try to improve plant growth, right? The objective is, can we change the spectrum of the light to match the plant stage and hopefully yield more yield, or at least energy savings? And there is examples of literature that have done this. For example, here you have literature that change the light through the photo period. For example, in the morning, they may have a different light quality than in the evening. But also you have fewer examples on how to change light at a different growth stage of the plant. And that's what we're trying to achieve here. So this is in general, the very general sigmoidal curve trajectory for plant growth. So you have your exponential phase, your linear phase, and your asymptotic phase. And you have your slope where you can consider having the highest relative growth rate. So the idea is, can we give different light colors at the different stages to maximize plant productivity, whatever productivity means. But as you can imagine, there is multiple options or combinations for this spectrum. So which spectrum to use and when to change. And there is a lot of research out there. My colleague Quinn Meng from the University of Delaware had recently worked on a lot of research using far red and green light and changing the spectrum. Also, another colleague in Michigan State, Dr. Eric Ranko, is working on the same. The approach that we decided to take was to look mainly at the red-blue spectrum and see if manipulating that can help us get better plant growth. So here, first, we started with a fixed light experiment where we didn't change the spectrum. We just grew the plants and collected the plants multiple times to the growth stage to quantify the growth. Here you have a summary here at the transplant stage and at the end of the growing stage. So we know that at transplant stage, 2080 red-blue spectrum was the best for overall dry mass accumulation. I consider dry mass accumulation as the most important factor for young plant production. In my opinion, even higher than leaf expansion and leaf area at this point. And then, so we found a high dry mass accumulation in both the 100% red and the 2080 blue. But of course, the 100% red had the red light syndrome where the plants were curled down. Then the later stage we find that greater percentage of blue, which is widely known again, have more anthocyanin content. But we also found that 100% blue was changing the architecture of the leaves. So we used this knowledge to create a dynamic recipe. So basically we had a light spectrum during the first 18 days of plant growth. Then we switched that light spectrum to increase leaf mass area and fresh mass. And then we switched that light spectrum for coloration, following guidelines from our colleagues from Michigan State. So here you can see an example of two of the experiments. So you have a fixed light spectrum where all the plant was run under 20 blue, 80 red for most of the time, except for the last few days. And a dynamic light spectrum that went from 20 blue, 80 red to 100% blue and finished in 50-50 blue red. So this is what we got. So the dynamic spectrum had 13% more growth in terms of dry mass and 25% bigger head diameter. Now, typically lettuce right now is sold by weight, has a wholesale, but also some other specialty products are sold by the unit. And sometimes the unit is correlated to how fast you can fill that clamshell. So in here, you just have a plant that has exactly the same fresh weight. So these two plants are holding the same amount of water. The one plant has a 25% bigger head diameter, but that plant has more dry mass. So these may be useful for some growers, indoor growers especially. Now we try to transfer this knowledge that we learn, which is the leaf angle expansion, to create a bigger head and hopefully more light interception into greenhouse conditions. However, we had a different approach. Instead of changing it through different growth stages, we tried to supplement the sun with signaling. Let me give you an example here. So here you have on the black rectangle, the hours of the day, 24 hours. Then a standard recipe, we call it, we had your standard fixture. In this case, it was a commercial fixture that had red, blue, and white dials. And we had that on together with the sun, which is the white, yellow spectrum in the back. And then we have we have that on through the day to increase the overall daily light integral. And then we had it, drop to 25 micromoles, and I'll tell you in a minute why, through the night. Then we had another treatment where we had the same standard spectrum and the first treatment through the day, but during the night, we only gave it 100% blue. Our hypothesis was if we can have that response of stretching the leaves with 100% blue and changing the angle, we may perhaps increase growth. Another one, and you see the justification here of the papers that we used to justify that treatment. Another one, you see the justification here, was to end with red light. After the sun set, we changed to red light, and then switch to blue light before sunrise, in hopes to increase the stomatal opening and be able to receive the higher radiation. Now we have a challenge in this experiment, and you may ask, why 25 micromoles? That's so low, and we agree, but this was a limitation of the technology. We couldn't give more of this specific spectrum unless we had a lot more lights, which we didn't at that point. We did this in an entire greenhouse, fully replicated experiment in 2 seasons. And this is just pictures of that work and effort. Let's just jump into the results. We did 2 different cultivars for that also. So these are the leaf area for the 2 seasons. We have a DLI of 20.5 moles in one season, another one of 17.1 moles, and you also see the difference in temperature. So we didn't see what we were looking for. We saw that, we have a leaf area that's slightly higher on the transplant stage, 11%, marginally, but we didn't see anything in the full size in the higher light or lower light season. So you can see here the standard, the finishing with red starting with blue, and the one finishing with blue starting with blue. You can see some difference on the transplant stage on leaf area, and but those differences did not translate to the entire growth stage of the plant. So basically, plant diameter was slightly higher, like we observed before, than your standard, but marginally. Right, so I wouldn't be giving any recommendations to anybody or using this recipe for a 5% increase in head diameter. But I think that goes back to the issue of our low light intensity during those times, that perhaps if that light would have been a higher intensity, we could have achieved the response that we wanted to achieve. Now let me jump into a different project that is also related on light characterization and impact of light on plant growth. So we wanted to characterize the impact of lettuce flowering and bolting, to characterize the light impact on that. So lettuce bolting, as most of you know, is a transition from vegetative to reproductive phase. And is in lettuce specifically, and many crops, stem elongation and flower formation is the first symptom. And that drastically reduces the quality of the lettuce. Let me give you an example. When you're producing lettuce in the field, in this case is iceberg lettuce, they go and sample the field and cut the heads in half. And any stem, this is the stem of a bolted lettuce, that is greater than 10 centimeters is not suitable for commercial, because the flavor of that lettuce now is different. So they sample the field and they find a percentage of the sample having a long stem like this is considered a bolted lettuce. They don't harvest the field anymore. So the bottom one is a bolted lettuce and the top one is a bolted lettuce. So we want to prevent that. Or you may be interested in rapid breeding cycles or faster seed production. Then is more important to actually increase or decrease the time to reach to that bolting stage. Now we know that bolting in lettuce is mainly controlled by temperature and secondary by photoperiod. So lettuce is considered a quantitative long day plant. It means the longer the days, the more suitable for bolting. However, most commercial cultivars have been developed to be a little less sensitive to the photoperiod, but they're highly sensitive to temperature, and temperature typically induces bolting. warmer temperatures. But light quality actually hasn't been investigated a lot for lettuce. So we decided to test this based on the previous research that we saw lettuce bolting. And we decided to test a knockout approach. Let's take some wavelengths out of the spectrum and see which of the wavelengths has the most contribution towards lettuce bolting. So here you can see this, I call this a control. You have blue, green, red, and far red, all of them more or less with the same ratio, right? And then you have one where we remove the far red out of the spectrum. And then we have one where we left the far red, but we remove the green while keeping the same ratio between the blue, red, and far red. And then you have one where we remove the green and we put just a little bit of far red on trying to see if we find a response with a little bit of far red. So this is another way to look at those treatments here. And basically here you have your PPF spectral distribution. And to make this clear, the far red was presented as a red to far red ratio here. So let's look at this plant. Now we grow these plants under very, very cold conditions because we didn't want to induce any bolting. We wanted to see if actually the light quality was enough to induce the bolting. So that's why you see this quite high number of days, because they were growing under very cool conditions. So at the beginning, if you can see the letters, by 18 days, which is the transplant stage, which there is a whole industry focused on lettuce transplant stage, those recipes, the pink one and the green one, that have no far red or very little far red, were doing better in terms of fresh mass growth. Then later on, those plants kept producing more fresh mass. And then things started to shift where the plants with the far red actually started having greater fresh mass at a later growth stage, right? Now this is just fresh mass. By the end, when the levels were quite high, they all have the same, but typically a product will be harvested around these days, so the ones with far red catch up, right? So this leads, for example, to another idea for a dynamic light recipe. We know far red at the beginning of the growth stage, changing to the additional far red later in the growth stage. But the whole purpose of the project was to look at the impact of bolting. So let's look at stem length in lettuce, which is one of those characteristics. So at the beginning, you have, of course, the plants with more far red with a longer stem. We know far red is a shade avoidant response in users, so the plants were stretching, and they kept stretching, and they kept stretching. But now at this point, the plant that has the green and the far red has even a longer stem than the plant that only has the far red, right? So it seems to be a synergistic response to the stem elongation when you have both wavelengths. You can see that here, and you can see that at the end. So obviously far red and green were elucidating that stem elongation. Now look at the plants when we harvest them. Here you have plants with the blue, green, red, and the blue, red, and low far red, with the stem size of these letters to be normal. And then you have those that are very, very bolted and long. But we also look into the leaves, into the plants. And here, if you're not familiar, these photographs are from the apical meristem, and they show how these plants are already on the flower initiation state. They have changed that shape of this dome. And then on your right, you have the leaf primordial, which they're still producing more leaves. So you have the ones with far red already changed to flower initiation, in those without far red or very little far red is still on the vegetative leaf production. This is how the plants look at the end. You can see the ones with far red and green are the most bolted plants, and then you have those with just far red follow quickly behind. But not only that, we also found that the allocation of dry mass between leaf and stem obviously was very different on the different plants. So basically the plants with far red had more dry mass allocated to the stem than those with without the far red or the very little red, you know. And this was consistent for fresh mass as well. So what we can learn from this is, number one, far red and green both have an impact on flowering. Far red is enough to induce it. There's still more research that had to be done to specifically find the synergistic interaction between green and far red, but they also affect the ratio of dry mass and fresh mass that is going to the leaves versus the stem. So to conclude this study is that we hypothesize that far red for lettuce is important or required for the detection of long days. And that conclusion may be a stretch, but all these plants were grown at long days, and the only ones that bold were the ones with the far red. So that's something that we can continue to investigate. So talking a lot about lettuce, and now I want to switch a little bit about young plants, and specifically of the use of light quality. Again, the use of those general understanding on plant responses to try to improve plant growth. In this case, we're going to look at tomato seedlings that either become transplants eventually, since every tomato plant, most tomato plants that are grown commercially start from a transplant industry and nursery. So the idea is, can we use light optimization to grow these transplants in a completely controlled environment to increase growth, you know, and have a high quality transplant that can move into the field, which all of that is good. Now, can include production, quality, and affordability of these plants, right? And however, typically energy and is one of the main cost and factors. So we can feed more plants in the same area and still maintain good plant quality, then the energy cost may be lower. So we had the question, why did we increase the density? Why we want to increase the density of these plants. You know, typically tomato transplants are grown around 400 to 900 plants per meter square in a greenhouse or in a high tunnel. What if we double that? What if we double that density to 2,000 plants per meter square? Obviously, if you work with plants before, there is going to be less light resources for the double amount of plants. So you're going to lead with probably lower growth and probably more difference between plants, less uniformity. So what we wanted to do is actually create a spectrum that can minimize the issue of the lack of uniformity. So in general, we did a preliminary study and we found that if you grow plants at 2,000 plants per meter square, you have between 10 to 20% plant loss due to the lack of uniformity. Basically, you have plants that are too small, that are lower in quality because they were competing, they fell behind at the beginning, and they never catched up. Now, if you translate 10 to 20% plant loss in some greenhouse cultivars where a seed may cost you 50 cents per seed, that's a lot. This may not be a problem if you're using, you know, more affordable seed like the one used for field production. But I see it has a problem. So can we use an ecological approach to use light signaling to increase that uniformity? Right? So for example, you have a plant here on your left that is exposed to the sun, right? It has equals amount of red to far red ratio, and the plant doesn't have signal to stretch or stays compact. On your right, you have a plant that is shaded by two different plants. So that plant is seeing a lower red to far red ratio, which typically consists of a light environment with more far red light in it, or more higher ratio of higher amounts of far red light. So the plant will actually start stretching. So can we use this knowledge at a trait level and see if we can achieve the same? So we decided to have a few treatments, and some of them are dynamic, right? So we have the days for plant growth until you go to a transplant stage, right? 18 days, around 18 to 20 days, you go and graft the plant, or you go and move it to a different spacing. So So you have 0 to 6 days, 6 days to 12 days, and 13 to 18 days. Under the right, the same blue to red light recipe. Then you have a treatment where we are a far red to try to elucidate shade avoidance responses for those plants that were shaded, under the canopy, those plants that came out later, and try to improve that growth. So we provided that far red from 6 days up to 18 days. Then we have one, which we're gonna call BRFR1, which we only provided the additional far red between days 6 and 12. And then we have another one where we provided a far red between day 13 and 18. So here you have some of the results. This is how the plants look, and there you can see 2 plants per little plot. So based on the tray size, this would get you a 2,200 plants per meter square, which is super dense for the production of seedlings. You can see the blended root quite compact, and then you can see the blue far red the whole time very tall, and then you see a differentiation in plant height between the far red provided at the between say 6 and 12 days, or 12 and 18 days, and you see the impact of that. This is so you have a total height of 197% taller if you have provided far red the entire time, versus 80% taller when you provide a far red at the beginning between 6 and 12 days. And then here you have your coefficient of variation, which is lower with the higher far red. So those shade avoidance response was actually working, but it was creating a very, very tall plant. Then you have stem diameter, which is very important for grafting. So you can see how far red the entire time increased the stem diameter of the plant, and all of it increases stem diameter compared to the ones without far red. And at the end, the dry mass allocation, here you can see the blue-red had lower dry mass at a 2,200 plant density, very dense canopy, than the one we have far red the entire time, right? And the same that they have far red from days 12 to 6 to 18, but the recipe that had far red from 6 to 12 days had higher dry mass than without the far red. In addition, you also show a good plant morphology or plant height, acceptable for growth. So we're getting close to the end of my presentation. I just want a couple of minutes just thanking my team. I get all the credit from presenting their results. So they're the ones that do all of the work. And I will open the floor for questions. Thank you very much.
Thank you, Ricardo. Very nicely done. We had a bunch of questions that came in from the Q&A, and so I'll read them off for you. So Rob's question was, is there a benefit to have different light intensities during a photo period? For example, lower during the start of the day, more intensity during the middle, and then lower again at the end of the photo period.
So we haven't done that research ourselves, but from other colleagues, they have measured the diurnal photosynthetic response to change to same intensity through the day and see how that decreases. So typically in some crops, the closer to the end of the day, the photosynthetic response per photo tends to decline. Now, you can take that one or two ways. You can decide to say, okay, it's declining, let's save resources and not provide the same amount of light intensity. Or you can take a different approach, it's declining. So can we provide more photons or perhaps more CO2 to try to give it a bump before the end of the day? But ourselves, we haven't done that research, but I think the diurnal change in light intensity and CO2 concentration can be a good strategy for improving growth.
Excellent. Thank you, Ricardo. Damon writes, great talk, Ricardo. I am curious about your opinion on whether natural sunlight is optimal for crop growth. Could natural sunlight be modified to improve photosynthetic growth rates?
Yeah, well, plants have evolved under my opinion, and plants have evolved under the sun to be successful under the sun spectrum. Now, success to a plant means reproduction, right? Set seed. If that's how you frame success, then the natural spectrum helps on achieving that success. Now, if we define success differently, for example, more fresh mass for a lettuce, more tomatoes for a tomato plant, or more lutein for a carrot plant, then I think changing the spectrum of the sun or helping supplementing the sun, it is possible to create a better spectrum for the end goal. We always have to go back and understand that sun is free. It's a free source of light, right? So supplementing the sun is often a good choice. Completely replacing the sun, uh, there needs to be additional gains on doing so, right? Additional gains to the plant because you are forfeiting the free part of the sun. Yeah.
Excellent, thank you. Someone asks, Kwame asks, what's the influence of dynamic lighting on taste?
Great question. Great question. So we didn't measure taste in this specific example. Um, so I couldn't speak towards that. But, based on, in this specific example, following, Owen and Roberto's advice on coloring the letters with the blue. So changing the phytochemical content of that leaf will eventually may transfer to taste. But there is some research done on taste and aromatics, by Kelly Walters and on basil. So there will be an impact. But on our specific treatment, we didn't measure that.
Got it. That makes sense. Someone asks, any reason why you didn't try a recipe with far red when you did the light recipes for greenhouse? Because it seemed to be promising under soil source conditions.
Absolutely, absolutely. Yes, far red is a very promising, and that has been shown. In our specific approach, we were working with technology, and still the majority of the light supplemental lighting technology is still focused on red and blue, and then you have a lot more shifting towards white, right? And then the addition of far red is starting to become more prominent there too. We focus on the red and blue to see we can provide a benefit to those growers that already adopted that technology that mainly has red and blue. But yes, far red it is a has it is a it has proven a benefit for plant growth. Yes.
Sure, makes sense. I found that in my own research program, it's hard to get good greenhouse grade far red fixtures, and sometimes we even have to build our own fixtures to do growth chamber far red work. Okay, good. Then Evan writes, really exciting results. Can you quantify the uniformity or decrease in loss in the tomato transplants with the far red treatment?
Yes, we did that, and I was shy of putting those results in the presentation because of lack of time. So far red, like the addition of far red, increase the uniformity. So and if the far red was provided to the entire growth cycle, it had a greater uniformity than those at the beginning or at the end. However, they have a coefficient of variation of, for example, the control had a coefficient of variation of 18%, while the far red had a coefficient of variation of 6%. So I was looking for a more impactful result, but even though it was significant, in my opinion, um, not yet granted the adoption for that use of uniformity with the results that we got. So that's why I decided not to include it.
Makes sense. And that's great. Uniformity is a really nice parameter to quantify benefits as well. We had another question, this one from Gabriel. You mentioned the effect of blue light on anthocyanins. Are there any effects of light quality on beta lanes? And I had to look up, these are like the yellow, red pigments that you get probably in like the beet family.
We didn't quantify that, so I couldn't speak to that. Yes.
Okay, so need for more research there.
That's right.
Job security for plant physiologists. Then Bob Hoffman asks, can you decrease tip burn in lettuce by changing the light recipe when growing in an indoor facility?
So I think from the stuff that I've seen, Bob, there may be an impact, but the actual mass flow of calcium through water, I think if you're able to improve the environment to create a good vapor pressure deficit at the growing tip, that will have a much greater impact than light quality on preventing tip burn. I know Dr. Cherry Kubota at Ohio State University is working on other ways to prevent tip burn. I found that the impact of light quality may not be as impactful as the movement of air around the tip or the movement of calcium up to the growing tip. So, but it can be, an additional layer. Let's say you already have a very good air management system and you still see a minimum amounts of tip burn. Maybe the next step may be looking into light quality. But how does light quality changes the mass flow of calcium through the leaf? Then we start, maybe working in stomatoconductance or so. But
That's good. Yeah, I was trying to think through that myself. I was thinking maybe if you had light quality treatment that gave you a more open head architecture, then you'd have more like transpiration through the growing point, but you still need the fundamentals of good airflow. Yeah. Mira asks, isn't it easier to control no sunlight than a combination of sun and dynamic lighting?
yeah, it's way easier to control no sunlight. You can do whatever you want, and any impact of your recipe will be directly translated to the plant, but it's more expensive, right? So if you're producing medicine using plants, or if you're producing young plants, or you're producing compact plants, or you're trying to overexpress a specific trait, maybe no sunlight makes sense. But when you're trying to produce tall plants with a lot of canopy, trying to provide all the needed light with only electrical lighting will be maybe cost prohibitive at this point.
Sure. Let me jump to one question that came in on the chat from Jorge. Jorge would like to know if it's possible to estimate radiation from LED micromoles, and to follow up, what he meant is from an LED fixture we can get the PAR intensity in micromoles per meter squared per second. But he wants to be able to estimate transpiration in a closed plant factory. So he needs radiation data. So he wants to be able to transform micromoles per meter squared per second to radiation in watts per meter squared. And I guess it can be done. It's just a matter of getting the right equations, and the equations are light spectrum specific.
Mm-hmm.
Is that right, Ricardo?
Exactly. And one way, a practical way, which is probably not the most accurate way, is to create a conversion factor using two different sensors. Like I said, it's not as accurate, but it may be practical. So you have a pyranometer and you have a quantum, and you can create a conversion factor based on your spectrum specific wavelengths.
That's a great suggestion. Yeah.
Practical, but I done that before when for my efforts outside the university, and tends to work.
Mm-hmm. Makes sense. I know for like sunlight, we have specific conversion factors. Even those are just estimates because the quality of light from the sun changes. So actually calibrating that with two sensors makes sense: a global radiation sensor and a quantum sensor for PPFD. Okay, then another question, assuming the PPFD for lettuce sole source is 200 micromoles with a certain spectrum, if you change the ratios of red, blue, green, far red, will it change the PPFD from 200 micromoles to less? And if so, how is that affecting the plants? So I guess you could talk about when you shifted ratios, did you keep PPFD the same?
Yeah, that's right. So in this specific experiment, the approach that we decided to take is to keep PPFD the same. So far red was always added on top. So two main goals that I had is keep PPFD the same, as is generally defined between 400 and 700, and we can challenge that in a different conversation. But and keep the ratios the same, right? So the ratio of the spectral present in that treatment should be pretty close to 1 to 1. So in this case, the PPFD was the same. And when we shifted between red and blue, the PPFD was the same. So generally speaking, on a fully canopy plant, photosynthesis of the whole canopy should be pretty close to each other. But any changes in morphology based on spectrum that will later translate into light capture may be different. But yes, in our specific experiment, we keep it the same. So far red was always added to. And we added a lot of far red. So right now we're working on the minimum amount of far red required to elucidate the response, which is ongoing research. Yeah.
Sure, makes sense. I know other researchers are looking at changing the definition of PPFD to also include far red. So, because there is some photosynthetic activity in far red beyond the morphology benefits that you talked about. So that'll be definitely we could look for those changes in years to come. Okay, then Richard says, excellent presentation. Some cannabis growers believe green light does not interfere with the photoperiod. Is this a myth, or is there a merit to this thought since green light can trigger shade avoidance?
We haven't done that test. I think green light will be absorbed by the plant and it will be affected photoperiod. We know some research that we did in the lab, we know that very low amounts of far red light, very, very low amounts of far red light are enough to trigger flowering in cannabis. So I envision levels of green light would do the same. But, that depends on the plant species. Dr. Eric Ranko for Michigan State University has shown how the plant responds to different red and far red ratios, and even to fluorescent lamps that don't have much far red, and that has become species specific. In my educated guess here is that a good amount of photon flux of green light will generate a photoperiodic response.
Yeah, and I know Michigan State University did some work with green light and bedding plants, and they found it could trigger a photoperiodic response, but a bit higher quantities were needed. So maybe if 2 micromoles of far red was enough to trigger the photoperiodic response. I forget the exact number they found, but something on the order of like 20 to 30 micromoles of green light triggered that photoperiodic response. I don't think they've done work with hemp or cannabis per se. Yeah. Great. Horace asks, have you observed any influence on the light spectrum on occurrence of edema on tomatoes?
Yes, Horace, we have done a lot of work on edema on tomatoes and young plants through light spectrum, right? So we know that the main issue is the absence of UVB, right? That's the main issue. And that can happen in a grow chamber if you're not providing UVB, or that can happen in a greenhouse if you have a strong enough filter and you don't have enough UVB going through the greenhouse. So UVB will fix the problem. Now, let's say you can't give UVB. Well, we have found that higher percentage of blue has a reduction effect. In addition, far red light also has a reduction effect. Even end of day far red light has a reduction effect on it. So when you combine high blue photon flux with end of day far red light, the time to reach the edema is significantly reduced, and the injury of edema is also significantly reduced. And that's in an environment with optimal humidity of 65%. Obviously, you have humidity higher than that, those symptoms will be more obvious. So high blue and end of day far red, or far red in general, will reduce edema symptoms on the plants, the tomato plants. Yeah.
Got it. Cool. Thank you. Then could you give an ideal experimental greenhouse light recipe, including far red radiation, photoperiod, PPFD, blue, red, and far red, compared to what you tried to focus in on with blue and red?
Yeah, I like this exercise. So I think I will have a recipe that focuses on dry mass for the transplant stage for few reasons. You think the highest loss of transplant shock comes from the plant being too soft and not having a good root system. Even more, I put that a higher priority than leaf expansion when the plants are very young, if they're going to be transplanted. So I focus with a recipe with high dry mass. Then I will, after you have an established seedling, I will start adding far red, to a certain point, perhaps into canopy closure, right? Once canopy closure, then I'll eliminate the far red, because then it will be, in my opinion, it will become detrimental by continue to stretch the plants. So after that, I will move to another recipe that actually has high photosynthetic response, and that can be white light, or it can be a good red and blue combination. Now, if you're trying to color, or if you're trying to create some phytochemical expression, then the recipe may be different. But if I had it my way, I have high dry mass at the beginning, no far red included in there. And then I move to a recipe that has more far red in it, and then remove the far red closer to canopy closure, and focus on light penetration through leaf angles. And yeah, that will be what I do. I need to try that myself first and see if it works.
That's great. I appreciate it. And so often people talk about light recipes, but your work is really nice. It's actually showing us some practical, beneficial light recipes, which is really great. We had another question that came in. Would far red be a good complement to a high blue light recipe or a high light intensity?
So for example, in the seedling work, high blue and far red is a great combination for multiple reasons. High blue tends to produce a very compact plant, which is typically what you want. But typically, when you're in a sole source environment, it becomes too compact, right? Too compact. And then, so adding far red to that, not only I just showed you that increase overall biomass, dry mass, it also alleviates some of that compactness and makes it more usable. Now, we don't want to give the far red through the entire time, because then you end up with a plant that is very stretched. So I think far red and high blue together, they balance each other. So I specifically like that if I can dynamically control the far red.
Okay, thank you. And then would it be doable to use filtered panels for the greenhouse to filter out unwanted wavelengths from sunlight while keeping as much free light as possible?
So light in the greenhouse environment, light is king. It's just like Vegas, cash is king, right? So if these panels are reducing the total amount of radiation going to the greenhouse, in general, there will be a yield penalty, and that depends on the slope. But if we go with the rule of thumb, you have 1% decrease, you may have 1% decrease in yield. So you need to be cautious if this filter selection will reduce your solar radiation, global solar radiation going into the greenhouse, which they often do. Now, let me give you an example where this can be used. If you're able to only filter out the infrared radiation, then you may end up with a more comfortable environment during the summer, for example, for a plant to grow, while not filtering out the visible radiation. And you can count it all the way to 800 nanometers that will be contributing for plant growth. So there is some technology out there called beam splitter mirrors. Still, they're producing a very small scale and too expensive to adapt to the greenhouse. But if we can do that, removing the heat or during the summer, or allowing it during the winter, and but still not mess up the visible radiation transmission, then you may have something in your hands. But splitting the spectrum, the visible, to make it more red, more blue, at a penalty of lower light, I personally wouldn't do that. I'll let the light in first.
Okay. And then someone asks, can you confirm that a general, not accurate rule for this conversion of watts per meter squared is 1 watt per meter squared full spectrum sunlight measured from a pyranometer is 2.1 micromoles per meter squared per second in the PAR range, which would be for sunlight only?
Well, I need to go back to my notes on those conversions. Yeah, I don't know, Neil, can you comment on that?
Yeah, I was just searching online and I found people asking this question. Mark Van Eersel from University of Georgia responded to one of those and did say the 2.1 conversion factor. So for each 1 watt per meter squared, decent conversion factor is 2.1 micromoles per meter squared per second.
There you go. I learned something new.
There we go.
Good.
Yeah. Interestingly, his response has in a growth chamber, and it didn't specify the lighting source. Maybe that was a fluorescent light in a old style growth chamber. The conversion factor was 4.6 micromoles per meter squared per second. So that's also a nice vindication that the light spectrum is very important for knowing the conversion factor. Okay, I think we've run to the end of the questions. I think you've hit upon a topic that had great interest and really nice to see the questions that came in. I'd like to thank our speaker, Ricardo Hernandez. Thanks for spending your afternoon with us, Ricardo. We really appreciate it. We do have an upcoming GLAZE webinar topic. The next one is on December 10th at the same time. And will be from Mirat Kachira at the University of Arizona, who's talking about dehumidification, ventilation, and airflow control in CEA. And we'll be able to post a recording of Ricardo's webinar on our website at glaze.org, glase.org. Thank you, Ricardo, and have a good afternoon, everyone.
Thank you very much. Bye-bye.